Next up, we've got Lynn Rothschild. She's a research scientist at NASA Ames and an adjunct professor at Brown University. She's a passionate astrobiologist focusing on the origin and evolution of life on Earth and elsewhere, while at the same time pioneering the use of synthetic biology to enable human exploration and astrobiology. Her talk is entitled Star Trek Meets ET, Synthetic Biology as an Enabling Technology for Space Exploration.
And in the spirit of diversity and inclusion, I'm a biologist, and there will be no equations in this talk. So I mean, obviously you have to start with, is human interstellar travel even possible? And the answer is it's inevitable. I mean, if we look at the, in the long-term perspective, ultimately the Andromeda Galaxy will reach the Milky Way in about 4 billion years. I probably won't be here at the time, but they will be merging about 2 billion years later. So we will ultimately have interstellar travel. I've actually thought that since the Earth will be uninhabitable well before then, we could launch Starship Earth, because I don't believe that anyone will want to be the one who makes the decision of who leaves planet Earth. And so I have pointed out that we could just launch. We could get a gravity cyst from Venus, pick up some CO2 there, which is fuel for the plants, pick up some water and CO2 on Mars, great water, in Enceladus, potentially, Europa, Ganymede. We could pick up some helium-3 in some of these gas giants, and so on, and wait until the sun becomes more habitable again, and then move back into the hood. So I'm assuming that that's not what you were talking about, though, that you want interstellar travel on a spacecraft, which is going to be extremely challenging. And so I'm here to tell you what biology might be able to do to help, because I can't deal with the fusion and the solar sails and stuff. That's the rest of you, all you brilliant people out there. So let me just stick with the humans. And as humans, whether we're on planet Earth or elsewhere, we have certain needs. We do need actually transportation. We need life support. We need food, oxygen, gravity, medicine, waste recycling, clothing, and so on. We need habitats. We need power. We need heat. We need light. We need radiation protection, which of course is even more important off-planet. We need communication. And just because this guy in Brooklyn was waving a wad of cash, I'll just add the fact that they're no Buck Rogers. But there are 4, as far as I can see, real stumbling blocks to interstellar travel. And let's go through those. Communication is severely delayed with Mother Earth. Or with really anyone else. And so that is going to be a problem, but I'm not going to really discuss that further. The time in transit is really a huge, huge problem. I mean, even talking about a Mars mission of 6 months in outside of Earth's protective magnetosphere is a real problem, and at low gravity. You talk to the astronauts about that. It is a real problem. Obviously, it would be a game changer if I could tell you that we could go on a field trip tomorrow afternoon to Mars and be back, in time to catch dinner in Montreal. That would be the game changer. Even if we got back next month, it would be fine. Even by Christmas, even by the next tax season, that's good enough. But the way we're talking about it now is going to be unbelievably challenging for humans. Radiation protection, of course, is another issue, and that's partly tied in with this time in transportation. And the radiation protection is really because we are carbon-based organisms. And organic carbon absorbs in the ultraviolet, but it also is highly damaged by ionizing radiation. And so it's sort of a non-starter for any organic carbon-based organism. And we could go into the astrobiology stick, and I would argue that we are likely to find organic carbon-based organisms anywhere we find organisms. So this is going to be a problem for life in general. And then, of course, we need this life support, which deals with the human physiology. I've alluded to it with the gravity and the radiation problems. We have a lifespan problem, and of course, the psychology, which I'm not going to go into because more people did yesterday. So the time in transit and the ionizing radiation tie in with this. So the question is there a technology that can help? And so I am here to provide that for you. So what if I have technology in this black box and I tell you that it's programmable? Well, I mean, so is everyone else's in this room. And say it's modular, which obviously is always an advantage. And it's self-replicating. And now wait a minute, we need this technology, we need it now. And it is self-repairing. How cool would that be if all your spaceships could even say those 4 things? But of course, this technology can do a heck of a lot more than that. And it goes perhaps without saying that what's in this black box, the technology that I've just sold you, is life itself. And so if you start thinking about life as a technology, there's all sorts of things you can do with it. So if you start thinking of life rather than this incredible diversity that nature has wrought, and this is my evolutionary biology background speaking, and instead is a micro machine that does carbon chemistry and nanotechnology, then, well, we used to say the sky's the limit, but I work for NASA, so that's just where we start. And so now all of a sudden, some of these issues might have some answers, maybe not for interstellar travel, but at least within our solar system. What about bio-based habitats? Or for radiation protection, what about drugs and gene therapy? For transportation, what about bio-enabled spacecrafts, nanotechnology, biofuels? And then for life support, aiding for the physical and psychological support. So what I'd like to do for the rest of the talk is basically say NIAC, 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 looking at Ron, who's usually there with the rotten eggs for me. A lot of this work has been funded by the NIAC program, and a lot of the proof of concepts has also been done by students in my lab on combined Stanford Brown University iGEM team, which is a competitive program in synthetic biology, primarily for undergraduates. So let's start with habitats. Now, obviously, the Apollo astronauts took their habitat with them, and as I like to say, it's sort of the turtle approach. You've got your habitat with you, you can count on it, but you waste an awful lot of energy carrying it around with you. And so that's what the nominal missions have, is to bring a habitat with us. But there's got to be a better way, and there have been suggestions of robotic construction of both regolith or ice houses. But what we've proposed is turning to the mushroom. Now, we don't actually turn to the mushroom. That's the fruiting body of a lot of filamentous fungi. But in fact, what we're turning to is the main body of the fungus, these hairs that grow underground called the mycelium. And that is the main part of the fungus. Now, others have discovered that if you take the mycelium and you put it in a mold, so yes, a mold in a mold, and give it something like wood chips to bind, you can now produce something with it. That's what mycelia are really good at. They're really good at binding materials. And so actually some of my students in the iGEM team in 2013 had the inspiration for making the main body of a biodegradable drone using this approach, which was kind of cool. And then we took that idea and ultimately turned it into this NIAC concept of using a fungal mycelial-based approach for making structures off-planet, something that we call mycotecture. So it's a portmanteau between mycology, the study of fungi, and obviously architecture. And if I start doing architecture speak, it's because we have 2 architects in the group. And this is an animation that Chris Maurer did, who is a fabulous architect at Red House Studios and has been a powerhouse. So our concept is that you would deliver this payload that has already been seeded with fungi on planet Earth, folded up. In this particular case, we're adding water that's sourced locally. This has also been seeded with algae. And the beauty of that is once we hydrate it, you'll see that the algae can take the CO2 that they source locally and the water and now start metabolizing using the sun as energy and produce oxygen. This oxygen is then available for the fungi to grow and bind. There's been drop stitches put in and other sorts of scaffolding because the fungi need something to go in it amongst, because if they don't, you end up with something that actually is very good dead ringer for leather. And there are a couple of companies that are using that approach. So if you see vegetarian leather, that's what they're talking about. It's a fungal mycelial leather. So anyway, that gives you an idea of what we've been working on. And obviously, because of planetary protection concerns and also the fact that you don't have stable liquid water on the surface of the moon or Mars, you have to have this in some kind of like double bag structure. And so we've turned to colleagues at, who do inflatables off planet, and you can see this fabulous inflatable that they've made for us, and obviously that's speeded up. Playing to the local audience, we also have colleagues at McMaster University, Michael Reinstadter, who has a wonderful planetary simulator, and so we've been able to test some of the materials in there. And that is all sort of airy-fairy, so let me show you that this sort of stuff works. So first, I'll just show you a little prototype stool that my students made a couple of years ago, completely rated for your average Stanford student. I can even stand on it without breaking it. They made this in about 2 weeks. It's a little bit spongy after 2 weeks. What you end up doing is baking it for post-processing, and then it becomes extremely hard. And I'm sorry, I wish I'd brought one of the bricks to show you. This is Chris Maurer over on the left. He's the principal architect at Red House Studios, and he's been just a powerhouse on this project. And you can see this 4x4 prototype that he's made, and this is when it was ungrown, and this was a few weeks later when the fungal mycelia have grown to cover it. So yeah, this is real. We can do things with this, and we don't even need to just do habitats. I envision that we could certainly do tables, chairs, you name it, with the fungal mycelial approach. So a very cool approach where you're growing on location. Now, what about transportation? Well, obviously you need spacecraft, but involved with that, you would want to have some access to nanotechnology, fuels, and so on. And once again, we turn to biology. Now, I know there are people in the room who are interested in mining, and in general, that problem with that is that you end up having to use large pieces of equipment. They're bulky, they're heavy, they're expensive, they're toxic. I was actually just in a panel at Canada Synbio 2023 in Toronto a few weeks ago on biomining. So I had to point out that actually in Canada, all members of the Canadian operations have to aim for sustainable mining. So they are interested in this kind of thing. And so this is what we get these needs, we've got these problems. Where does life come in? Well, think about it. Your body is not just made of organic carbon and water. We use a lot of metals. And I always tell people, if you're interested in what metals, look at the back of a vitamin bottle. That'll give you a head start. So because we need metals, we have to somehow use them. And we can't just sort of randomly use them. We have to have great specificity. So, for example, your hemoglobin can't just wake up one morning and say, I wonder what it would be like to use cobalt. Or wouldn't it be fun to use platinum today? It doesn't work that way. It is iron every day, all the time, or you're dead. So what if we could harness that kind of specificity and apply that to metal extraction and separation? And so we've done that, combining that, and again, thanks to the NIAC for a phase 1 on that, we've combined that with some of the mycotexture work. And so what you see here is a micrograph I took of the fungal mycelia and then a cartoon drawing of silver atoms attached to that. So what we did is we took fungal mycelia, we made peptides, so short proteins that could bind to the outside of the fungus, as well as very specifically bind to, in our case, copper, and created a functionalized mycelium. So what you've now done is created a metal filter for filtration, separation, remediation that you could grow into any conformation, and in principle is completely biodegradable at the end of the lifetime and completely tunable as long as you can have a peptide that binds to whatever metal you're interested in. And we spent part of the COVID lockdown actually combing the literature and have a fairly good library now of peptides that have already been published that will bind to very specific metals. So we don't have the entire periodic table filled in, but we've got a good head start. So what about nanotechnology? You hear the word and you think of a clean room. Well, Simon Vecchione was the captain of our 2013 iGEM team. And what he wanted to do is build a radio transmitter with using the SETI definition, meaning he was the only intelligent person in the lab. And I couldn't build a radio transmitter with a kit from, the dear departed Radio Shack. So we talked him down off the ledge. And what he ended up doing is finding out that if you take a DNA double helix, and instead of having cytosine bind to guanine, which is typical, but you force a cytosine-cytosine mismatch, there is now a gap between the two strands. And what's really interesting is silver very specifically goes in to fill that gap. And by the way, thymine-thymine mismatch, you end up with mercury. So now you not only have a way to biomine, but you also have a way to make a completely tunable wire that is the thickness of a double helix of DNA. DNA is really cheap to make. This is really easy to do in the lab, no clean room needed. He went on and did his PhD with NASA STMD funding at Columbia University and is now a postdoc at NYU, continuing to develop this into 3D origami and so on. In fact, this is a recent NMR he did. It turns out that it's not just one silver atom per mismatch, it's 3. So again, completely tunable. You could have mismatch, mismatch, clamp, mismatch, mismatch, clamp. You could do anything you want along this either two-dimensional wire or then, again, with the three-dimensional. Now, years ago, I was talking to the head of Armstrong Center at one point, and he said to me, do you think you could ever have an organism take chemical energy and convert into electrical energy? And I just looked at him and tried not to show my amazement. I mean, that's actually what we did by eating lunch today. Anyone who is still alive in this room has taken chemical energy and converted into electrical energy. Well done. It's just that the electric eel is very good at storing it and releasing it like that. And so obviously life is very good at that and in principle could be done. Biofuels is something we've stayed away from in my lab because the commercial sector has been very interested in sustainable aviation fuels. And as a phycologist, someone who's trained working with algae, I'd like to point out that the first flight with a different blend of sustainable aviation fuel was done with an extract from Botryococcus brownii, which is a green alga. So, again, all sorts of ways that life as a technology can help. What about radiation protection? Well, gene therapy, drugs are things that come to mind immediately. And so once again, thanks to the NIAC program, we proposed that there is a way to solve some of this problem of drugs on demand. Now, you obviously cannot take an entire pharmacy with you. You don't know who needs what. You don't know how much they're going to need of everything. And even worse, it turns out that there are a lot of drugs, particularly ones called biologics, that are not shelf stable. So even under refrigeration, many of them are no good after about 6 months. So it's not even going to get you on a round trip to Mars unless all of you solve the 1-week field trip propulsion problem. So what we did was we proposed a miniaturized on-demand production system for biologics, which could be extended to non-biologics eventually. Once again, a cast of 1,000. We are really grateful that that's also just gone into phase 2. And the basic concept is here that we program Bacillus subtilis. It's a particular bacterium that's found in the soil, incredibly resistant to radiation and the elements when it's in the spore form. And I'll show you a picture of that a little bit later. In fact, it's been flowing on LDEF, the long duration exposure facility, for nearly 6 years, and some of them survived. It wasn't intended to fly for 6 years. That's just how long it took to pick it up. And so the idea is we've got these Bacillus subtilis spores that are pre-programmed to make a huge diversity of drugs. And by the time you dry them down on a little tiny hole punch of paper, it weighs nothing. It's virtually massless. And so you can then literally take a pharmacy with you. You then activate it by adding medium and you do some purification and you're ready to administer the drug. And we're aiming for 24 hours with little kits about the size of a wallet. I could go through that in detail, but it turns out that the Daily Star beat me to it. Yeah, I was not expecting that. And it's sort of funny for the people who are British in the room. The BBC picked it up from the Daily Star. I thought it was supposed to be the other way around. Fun fact. Okay, but we could also, as I mentioned, gene therapy, we could start to look to other organisms that have solved the problem of living in extreme environments. And for that, the project that we've had going on off and on in my lab since the iGEMer started in 2012 takes advantage of the fact that my interests for many years in astrobiology with life in extreme environments. Well, these organisms have the capability, and if you start to think of life as nature's hardware store, why not take some of these capabilities and put them in another form factor, including potentially using them to give us a leg up in some of these environmental extremes? I'm very fortunate that I have a relatively new postdoc in my lab that has now picked this up again with, instead of one gene or operon at a time, the way the iGEMers did, is going into entire environments and pulling out whole groups of genes with functional metagenomic screening and also adaptive laboratory evolution. I wanted to tell you this. I don't know if you recognize this picture. This is Octopus Springs in Yellowstone National Park. It's an old picture. My daughter, who's there at about age 4, just got her PhD. So it's a very old picture. But if you turn your head a little bit, it looks kind of like an octopus. And that's why it's called Octopus Springs. Now, fun fact, it's about a 5-minute walk from Mushroom Springs, which is where Taq polymerase was first isolated from. And it actually, Mushroom Springs looks very much like this, except it obviously looks like a mushroom instead of like an octopus. So there you go, fun fact. There is also a group of people around the world, primarily in Europe, Japan, and the United States, who are focusing on building cells from scratch. Now, NASA's funded origin of life work for many, many decades, but this is not only that kind of work, but also looking at the art of the possible. So not necessarily trying to follow the steps that we think that life took on planet Earth, but what could you do to make life from scratch, either from top down or, much better, a bottom up sort of approach. And now you could imagine forming a life form that is much more resistant to radiation and so on. I know chemists who are far better than I, and I say, well, look, this is a problem with the DNA double helix, for example. And they look at, they say, if we take out this double bond and we move this over here, so, and then you will find something much more radiation resistant. So, again, there's all sorts of potential if we start bottom up. Anyone in here who is at all interested in this, there's a Monday seminar series that Kate Adamala at Minnesota runs. They're all online, Build a Cell. They're spectacular. I am just, in awe of the kind of work that people do all over the world in this. Life support is an obvious one. I've already sort of hinted a little bit about some of the life support possibilities. We're organic carbon-based organisms, we eat or other organisms, obviously life support, and we breathe oxygen produced by other organisms. So it's very obvious to use these kinds of approaches in space. We're already really good at recycling water. Yes, today's coffee is tomorrow's coffee, and so on. But beyond that, again, why not have organisms do chemistry? You do not need to, have an off-planet account with sigma. You can again turn to life, because it turns out that life is really good at doing chemistry. In fact, many molecules in the Sigma catalog are biologically produced. And so I show this picture, and I think that pretty much says it all. Life is also a material in itself, and I love to show pictures of Scottish sheep because it reminds me of the wool and the leather. And I try to get the trees in the background and so on. Life is a material, and so we can certainly use that, as I mentioned, with the fungal mycelium. But the hardware store that nature has provided, unfortunately, is not ideal for shoving in a spacecraft. We're not going to take sheep with us, no matter how cute they look, and we are not going to take fields of cotton or rubber trees. Someone asked me at NASA headquarters once if we could program an organism to produce rubber. And I looked at him in disbelief and I said, you do know that rubber actually grows on trees, right? And this is how I become persona non grata at headquarters. Anyway, so there's the rubber tree. And silkworms, were not likely to take those, although I have Japanese colleagues who have suggested that making kimonos on a long duration flight to Mars would be a good way to occupy astronauts. However, We are likely to take these. And again, this is a much bigger picture of Bacillus subtilis. And here you can see the white dots, which are those spores that I alluded to before. And so again, really resistant. These spores have proven themselves in space. And when I flew a space mission, which I'll talk about in just a second, we went to Bacillus subtilis spores. Not because I think they're cute. I'm a protozoologist by training and love. They've got much more personality. But by golly, these guys let you sleep at night. I mean, they are bomb-proof. So why not take those and just take nature's hardware store and put it in a better form factor? And so that is exactly what we're proposing. That is what we're doing with the astropharmacy and to some extent, secondarily, with the mycotexture work. So if you do an analogy to what goes on planet Earth, you can take these raw materials that are available on the planet, which is what life has done here, like water, carbon dioxide, light, and minerals, convert those into organic compounds that animals like us can eat. And then you potentially off planet could, or on planet, take those and then bioprint them and make materials that have never evolved naturally and that could be amazing. I mean, imagine if you had metal inside a 5 micron circle of cotton, bound by, whatever you've programmed your Bacillus subtilis or your algae or whatever to produce. It could be amazing. And once again, we had a NIAC on that. And once again, very grateful. And we do have a patent out on that as well. And this last point I'm going to make may seem kind of trivial to people in the room who are molecular biologists, looking at the molecular biologists in the room. We will send digital information to make DNA. This is true massless exploration. Up to this period in time, there has been a physical continuity between the origin of life and ourselves. But we have now broken that continuity in the last few decades. So now we can send digital information. Our students do this every day. They go into the computers, they make up a DNA sequence, they push send, submit. We put our credit card information in as the PI. It goes over to some DNA synthesis facility, and back comes the physical manifestation of your DNA. It doesn't matter once you've broken that link whether the facility is across the hall or on another planet or in another solar system. You have broken that physical link. And so obviously this could lead to a whole other talk about how you could program to make life elsewhere without actually physically taking it, as long as you had that infrastructure to build things. So I've given you a lot of wild and crazy ideas, got many more up my sleeve. But at some point people say to me, well, this is great for the distant future, but what could you do now? Well, we were very fortunate in 2012 to be selected to be a secondary payload on a DLR, German satellite called Eucropus. Now, Eucropus was short for Euglena, which is beautiful little protozoan, which we all used to learn about, but I'm afraid kids may not have seen it. But look it up online. It's a beautiful one. Crops in space. And so the idea was to have a urine recycling system to grow tomato plants. And their basic concept was this satellite would start off without spinning, so it would be mimicking microgravity, and then spin it to mimic lunar gravity, about 6 Earth's gravity, and then spin up to mimic Martian gravity. So that was kind of cool. I would have preferred to test radiation, but it was a great opportunity. I've worked at NASA for decades, and people don't just walk up to me every day and say, how would you like to fly? So it's like the only answer is yes. The only sad part about this is that the Germans lost contact with their payload, so they didn't get anything out of it. So at some point, we were the payload, but we did. And obviously, I didn't do it by myself. Cast of 1,000, both in my lab and many fabulous engineers who were involved, both NASA and the German side. And so I was told to come up with a payload that could be as cheap as possible. Therefore, we reused some microfluidics that had already been flown on a small sat, and to involve synthetic biology. Now, I gave you that concept of organisms being that interface between the raw materials on the planet and the organic matter that's used to feed other organisms. This is a concept that my students called Power Cell in 2011. So it's sort of the biological power center of a colony. And here on Earth, that role is taken up by cyanobacteria, algae, plants. In space, we're imagining that this is likely to be something like cyanobacteria, some of which can also fix nitrogen, and algae. So that's the power cell. That's a filamentous cyanobacterium in there. And so we tested this concept of whether you could have these cyanobacteria then feeding the Bacillus subtilis in space. That should have been a no-brainer. We had 3 different gravity regimes to test it. I went up pretty much agnostic. If there was no gravity effect, fabulous. It would save the agency a lot of money. We could go back and test things in the lab. If there is a gravity effect, we should know now and be able to account for it. So there was the power cell aspect. We did a transformation in space, which is like really fundamental part of molecular biology, getting new genes into an organism. And I should add that's usually done in labs with something like electroporation. And if you want to see an engineer faint, say that you're giving them a shock of electricity. So we actually ended up having to do something kind of funky with the strain that was made competent by using xylose, which turns out sort of tripped us up in later experiments. And we also tested promoter strength. And to cut to the chase, because you don't want to see a lot of data, power cell worked great. The transformation worked on the first 1 or 2, but it turns out you can't leave xylose in LB for several years. Who knew? Because it ultimately caramelizes. And the promoters, I'm giving it a sort of a blank because certainly we got gene induction, but the one that I thought would be stronger than the other was not. So final thought, is human interstellar travel even possible? You know, get us the propulsion, please, everyone else in the room. But we can help you part of the way there once you realize that life is a micro machine that does carbon chemistry and nanotechnology, and that this micro machine can be even more useful if you start to add in synthetic biology. And perhaps this ultimately can overcome some of the limits of carbon-based life. And if I could just have 1 or 2 more seconds, my thesis advisor, Annette Coleman, who is a wonderful phycologist, passed away during our opening on Monday. And so in her memory, I'd like to say ad astra. And thank you very much.
We have time for one.
My watch stopped in the middle of it.
We have time for one question.
Thank you. That was a wonderful talk.
You are.
My question is, if we want to build sustainable environments, let's say on Mars or the Moon, and if we don't want to rely on things like fragile systems like hydroponic systems, it seems to me we're going to have to develop something akin to local soils. So what would your suggestions be in terms of how to bootstrap soils for growing agricultural products or even just plants to produce oxygen?
I don't normally deal with multicellular organisms. I'm not quite sure why you immediately discount hydroponics. I'm not really sure what you're getting at, because I could see ways to go at it. Part of the problem is that the lunar regolith is very sharp, and the Martian regolith has a lot of peroxides in it, perchlorates, and so we are actually thinking of ways that we'd like to get at to detoxify the perchlorates. There are organisms that can do that, and therefore make Martian regolith more tractable. Sort of as a side note, which doesn't answer your question at all, but we have used lunar and Martian regolith simulant with the fungi, and as long as they've got a little nutrients in there, they've been fine.
Very much.